welcome everyone. It is my pleasure to welcome you to the GRC Connects on Preclinical Form and Formulation for Drug Discovery. This is the second event we've had this week, and I want to thank those of you who attended on Monday and who have come back on Wednesday, and those of you who are just joining us for the first time. As you all know, we couldn't meet in person this summer. We were supposed to be in New England, where I'm sure it is beautiful right now. But I'm happy to see all of you here in the Connects feature, and we will see you in 2023, hopefully, in New England for an in-person meeting. So I just want to introduce myself. I'm Julie Champion on behalf of my co-chairs, Annette Bach, Erica Schlesinger, who is going to be co-chairing this session with me today, and Lynn Taylor, as well as Leah Burson and Matt DeFrez, who are going to be running the career panel that is taking place later this session. So without further ado, I would like to introduce our keynote speaker, Professor Maria Croyle. She is a professor of molecular pharmaceutics and drug delivery at the University of Texas at Austin. And she has really a long expertise in developing vaccines. For example, she led an international team back in 2008 to develop needle-free Ebola vaccines and was really the first to report long-lasting protection after a single dose in non-human primates. And some of her methods that she has developed for stabilizing live vaccines have now been patented and are licensed by a startup company, Girata, in the Research Triangle area. She also has served as an expert on the USP Biologics and Biotechnology Expert Committee for cell, gene, and tissue therapies, given her expertise. So I don't want to say much more to give her the time to present her work, which I think you're all going to find fascinating. So don't forget to put your questions in the Q&A and upvote them if you think they're exciting. And welcome, Dr. Croyle, and thank you so much for presenting to us today.
Thank you so much for that introduction, and thanks to the organizers as well to allow me to come and talk to you guys about some of the work that we are doing. Let's see. Okay, so the work I plan to talk to you today about is work that started here at the University of Texas College of Pharmacy and is the subject of a small biotech company that we've spun out called Girata. So this is my disclosure slide. And so really where this concept actually came from was when we received funding, or actually when I was contacted by Heinz Feldman and Gary Kobinger, picture here on the left, to work on a project they were spearheading to develop a needle-free Ebola vaccine. They had significant experience responding to Ebola outbreaks and knew that they needed something easy to transport, administer, something that would be easily acceptable to a variety of people and relatively low cost. And again, something very easy to administer, despite the fact they did have a strong support system on the ground, but also something that could be used in remote villages as well. So the platform that we decided to use was an adenovirus serotype 5 based vaccine that expressed the Ebola glycoprotein and did it actually quite well. The targets that we were looking at was to look at a nasal administration route or product and something that could potentially be administered orally. And one final issue that we needed to address that's very well known within the adenovirus field is this concept of pre-existing immunity. A significant portion of the general population has pre-existing immunity to adenovirus serotype 5, but the population we were really initially developing this vaccine for had the most significant level of this as well. So this was something else that we added to our criteria to look at. So this work was conducted by a wide variety of students in my lab, where first we were trying to develop a formulation that really could allow the adenovirus serotype 5 to override pre-existing immunity and neutralizing antibody in a living system. So first what we did is we conducted in vitro screening assays to look at the ability of the virus to infect cells with in the presence of increasing amounts of neutralizing antibody. Once we found that, we then went to looking at the ability of the vector and the formulation to foster transduction efficiency in targets. And here you can see in this bottom panel that when administered to the lung, the formulation that we identified called F16 really did support significant amounts of transgene expression indicated by blue staining in naive animals and also those with pre-existing immunity as well. We looked at, again, criteria. We needed to improve the systemic T cell response, and this formulation also did that. And also we saw T cell responses in the mucosal area, again in the lungs, so systemic T cells and lung T cells, an improvement in the antibody response. And one thing that we found really specific to Ebola is that we could increase the amount of polyfunctional T cells in response to Ebola glycoprotein with this formulation as well, something that was compromised by pre-existing immunity to adenovirus and which was also needed for survival. But I won't lie to you, this wasn't something that we just came up with, really quick in the lab. We screened over 4,000 formulations before we actually found this particular one that really seemed to work. On the oral delivery front, we were also looking at the potential of, what could we do with respect to administering the vaccine by novel routes. What you can see here in the middle panel is the immune response we achieved by just simple oral administration represented by PO. And you can see that was relatively a very sad response. We didn't see anything. But when we decided to place the vaccine in the formulation under the tongue, we saw a significant immune response represented here by the SL group. It was comparable to what we saw by intranasal administration, and we were really excited about that. Again, with the fact that we could also get around this pre-existing immunity issue and really see a strong polyfunctional T cell response as well. And we also did some histology, and we saw that macrophages, really, and dendritic cells were very good at coming in and collecting the virus in areas of the sublingual mucosa within 2 hours after administration. If we looked at 48 or 24 hours after administration, they were gone. And again, the data really led us to believe that they took that antigen back to the T cells and were capable of inducing a systemic immune response. So with that, we then thought, well, how could we actually, do this in a way that would be easy to administer in developing worlds? And so this kind of, led us to think about, is there a way to take that optimal formulation and create a dosage form in which we could effectively trap that vaccine, again, a live recombinant adenovirus, within a film matrix and use that as a means to deliver the vaccine by the oral route. And so we did do that, and over time we had notable stability at room temperature where we embedded this vaccine within the film matrix for 36 months. So essentially 3 years, we saw very good stability. And we were also trying to think about, again, with the nasal platform, what if we took that rehydrated film and used it as a liquid, potentially as a nasal spray? And in that particular case, we saw very, very solid stability as well for up to 8 months. Again, this is rehydrated film. So with the Ebola project, the proof in principle really was to immunize animals and expose them to Ebola. This required room reservations in a biosafety level 4 facility. And by the time we got to primate studies and were ready to really test this formulation, we had a good grasp on what the dose was for an effective protective dose by administration of the nasal route. And so what we decided to do with this film was to actually send the film into the East Coast where the primates were housed in a FedEx envelope. So no dry ice, no cold packs, just an envelope with some film in it. And then we went there, rehydrated that film, and administered the vaccine to animals as a nasal spray. And what you can see here, this is a very busy slide, but really the survival curve kind of says it all over here. What we also administered in this particular study was we used what was considered a 50% effective dose. So you can see the survival, the orange survival curve highlights this. So this was the same vaccine that was not in our formulation. It was actually shipped on dry ice with standard PBS glycerol as the formulation, and we achieved that 50% survival dose when that was given to animals. If we took that same frozen formulation that did not contain our film matrix at all, we did see a slight improvement in survival when it was administered by the nasal route, but our rehydrated film and administered as a nasal immunization in these animals allowed 100% of those animals to survive. So we were very intrigued by this, but we didn't want to stop necessarily with considering the use of the platform as an oral delivery device. So we went back and did some studies in human buccal explants. In this particular case, we used a live influenza virus embedded within the film matrix. We placed these films on buccal explants and then over a period of 3 hours looked at the ability of the live virus to transit through the human explant and be retained or assayed in the basolateral chamber. So what we looked at here was what we considered to be a high dose and a low dose of vaccine, and then obviously the unformulated control. So what you can see really in the right panel here kind of summarizes everything up. With the unformulated control, all the live influenza virus was retained within the explant, but never transversed to the other side of that tissue, the basolateral side. Whereas with the high dose and the low dose, we definitely saw significant amounts of that virus being able to transverse across the entire explant and be detected in the basolateral media. There was some virus that remained within the tissue, but that was also something that we kind of expected, but we're impressed definitely that the film matrix really fostered transit across the buccal mucosa. This then led to one of our first proof in concepts of really using this as an oral administration device. One thing to think about, and this was the other significant hurdle that we had to achieve, is we had to scale down the film to the point that it would be able to easily fit under the tongue or in the upper cheek pouch for buccal administration of a mouse. And so what's shown here actually is not the representation of that actual film. This would be equivalent to a 1 milliliter type solution dried down into the film. We actually scaled down that we could dry 100 to 150 microliters of a solution into a film, and that's what we used to place to dose these animals. So in this study, the treatment groups were an IM administration to reproduce exactly what's done with the standard flu vaccines today, and this was unformulated. The other vaccines were embedded in our film and either, as I hinted at, was administered by the sublingual route, placed under the tongue, or the buccal route, placed in the upper cheek. And then the last group that we did here was we kind of threw care to the wind, I guess you could say, and we took films with live influenza and stored them at 60 degrees Celsius for 4 days, and then administered them by the buccal route to see what types of immune response we would achieve with a significant excursion from just room temperature conditions. And the data that we received from those animals is summarized here. In the left panel, we simply looked at the amount of systemic IgG, anti-influenza IgG, presented or produced after each route of administration. And so what you can take away from this is that there really was no statistical difference between the different routes, and even including that those animals given the film that was stored at high temperature for 4 days. I'm always, the skeptic in the group and says, well, that's great, there's IgG there, but is it really neutralizing? The answer came back as a resounding yes, and that's what you see in the central panel. And so we were very excited about that as well, that very strong immune responses across the board. We saw more variation in the group that received the films that were at 60 degrees Celsius for 4 days, but still we were very impressed with the level of neutralization we could achieve. And this was, again, systemic neutralizing antibody response. When we went back and looked at systemic cytokine patterns after immunization, it seemed like each of these routes of administration really favored the Th1 response, which indeed we believe could perhaps help with prevention of COVID-19 for sure. Just to show you as how adaptable this particular platform is, this isn't necessarily used for vaccine, but we did have a group at AskBio ask us if this could be used as an injectable product. This is definitely not what we initially intended it to be used for, but we are always up for a challenge, so we said, why not? The optimal formulation with the data that I've showed you up to this point has a very high viscosity, kind of around the consistency of maple syrup. So we tested various formulations at different viscosities to determine if we could lower it enough to be acceptable to be used as an intravenous product. And we also determined the limitations of the technology as well. So again, with formulation 7, we see that we didn't get as much active AAV back from the film after drying, but this was also a very difficult film to form. So again, this was really our 100 centipoise was really our cutoff with respect to viscosity. And so that is the formulation that we decided to evaluate with this group for use as an intravenous type product. The stability profiles with AAV was also very strong and robust. The red bar in each panel was the current marketed formulation that AskBio uses for their AAV products. And the panel on the left represents 4-degree stability. And so again, very strong stability for up to 30 days. We've taken this out to 90 and 120 days with the same response. And what can be shown here is that, again, the AskBio formulation has good stability as well at 4 degrees, but our formulation seemed to show a bit improvement with respect to transduction efficiency. What you see in the right-hand panel is what we offered their formulation with respect to room temperature storage. So again, looking at the red bars, we saw a significant decline in active AAV in their liquid formulation when stored at 25 degrees Celsius, with the activity in the film being quite high. So the next thing that we decided to do was, again, test this, evaluate the ability of the films to be used as a product. So essentially what we did, we had tested 2 formulations. Our original high viscous formulation, where we made it, we embedded in the amount of virus in the film 10-fold, so 10 times the target dose, so they could rehydrate this in a different volume, and then our low viscosity formulation as well. So again, these were stored for 30 days at room temperature before sending them to AskBio in North Carolina in an envelope. Again, they were quite amazed that there was no dry ice there or anything with those, whereas the control vectors, again, they're fresh formulated virus we sent on dry ice, and then one aliquot of that had a room temperature excursion for the time it took us to form the films. And this was a luciferase expressing virus. It was an AAV9 serotype. I don't know if I said that before. And basically what we saw from these animals over a period of 29 days is that, again, group 4 and 5 were the animals that received either the high viscosity or the low viscosity film. The levels of gene expression in the liver really were not significantly different than the fresh virus controls in their original formulation. When we did biodistribution analysis at the end of the study, we really saw across the board, if you look at the last 2 bars of each of the clusters responding to different organs, there really was no significant difference between the fresh formulated virus in their standard formulation and that in the films that were stored for 30 days at room temperature. So we were very excited about that. So you might be thinking, well, how does this work? This sounds really, interesting. And so we did have performed some studies, largely with adenovirus at this particular point, where we have really gathered some data showing that there is specific binding of particular excipients to the adenovirus capsid. And this is just a summary of FTIR analysis that was published in a paper last year in Science Advances, whereas we added stepwise, several excipients to the film matrix. And we looked at, patterns with respect to adenovirus. We saw patterns that suggested that there was nitrogen bonds being formed between the virus capsid and the particular excipients as different things were being added to the platform. The graph on the left represents a really elegant study performed by Ornella Badjovic, who was a graduate student in my lab with the adenovirus. And what we, after looking at the FTIR data, we suspected that the excipients were stabilizing the virus by binding to hexon proteins. And so what Ornella did in this particular example is looked at the stability profile at room temperature of the virus in the film matrix, solid over 25 days. But when she titrated in a particular amino acid that would interfere with binding to what we thought would be the hexon proteins, the stability tanked within 3 days. We were really excited about this result and very intrigued. When she added another amino acid that didn't interfere with the proposed binding, we didn't see that response. And so this is the subject of a paper that's currently under review in the Journal of Controlled Release. When we looked at our work with AAV, we wanted to look at the 3 AAV proteins and how they changed or didn't change over time when embedded in the film matrix. And initially, we were very intrigued when we ran the entire formulation on an SDS page gel. And you can see that here in lanes A and B, that this represented the virus in the full formulation. And so it just seemed to be this blob right here. And so we had to actually go through a very detailed heating process to actually release the capsids from the overall film matrix formulation. So here is film without virus in, and again, D represents the heat treatment that we did to actually get the capsid proteins to be released from the virus. So we really do believe that this is how the virus is stabilized within the film matrix is that the film components really lock the virus in and protect them from any kind of extraneous outside stressors. We've done a bit of work with antibodies, and this represents a work that we collaboration we had with Jennifer Maynard here at the University of Texas at Austin, where we took some of her humanized anti-pertussis toxin antibodies, embedded them within the film, stored them at room temperature for 30 days, and came back and just evaluated their ability to bind to the pertussis toxin. And each of the films that we used really did have retained that binding capacity. And we also looked at the impact of rehydration on aggregation, and it didn't seem to have that type of effect at all. So the last thing I will share with you is some work that we've done with nucleotides. Initially, we started this work several years ago prior to COVID, where the concept was really looking at more of a manufacturing process and maybe not necessarily delivery at this particular stage, where you could take your plasma, make a recombinant virus or some therapeutic protein in a bioreactor.
Hi, Erica. Yeah, it must be Maria, not us.
Okay. Did we just lose her entirely?
I don't know. I wish that the GRC staff would. Yeah, Megan is saying, yeah, it was her. Hopefully she can just join back in. Everyone can hold tight for a few minutes while we try to get Maria back in. She was almost done. I sent her an email. Okay, so Megan has asked us if we could keep talking. So maybe there's been a lot of questions. So
yeah,
I wonder. Certainly, we are not qualified to answer the questions, but there might be some aspects that we could talk about. I thought that it was interesting. Oh, here she is.
Yeah.
Hi, Maria.
Okay.
Go ahead and finish up, and we have lots of questions when you're done.
Oh, okay. Well, that was pretty much it. Definitely it's a technology, it's a platform technology, and I just wanted to do the pitch at the end. So with Gerada Thin Film, we are hiring, and I know you guys are going to have a career panel later. So we're looking for scientists of a variety of different levels to come help us break the cold chain.
Wonderful. All right. Well, thank you very
Great.
much.
Well, thank you, Maria. Sorry for the technical difficulties at the end, but as Julie mentioned, we have a lot of really great questions. So hopefully we can pepper you with them and get some responses. So I think sort of the top of the list for questions was regarding the drying technology that was used to dehydrate the film. And I might just expand on that question to say, could you speak a little bit more to the production process in general, as well as the formulation of the films and the types of excipients that you're using?
So that's a really good question. And I realized as I was putting this together last night, I didn't really talk about that, but it's really because the process is simple. It's not lyophilization. It is essentially, we prepare these things under aseptic conditions, and the drying process takes place at room temperature. So it's just under airflow and no real forced anything, no vacuum, no tricks. Essentially, it's just essentially air drying, if that makes sense. And really what we've developed this technique for is we can formulate in the morning, lay these films out, and by the end of the day, peel and package. So it's really a nice system. And it really, again, was for developing countries. You don't need much electricity requirements. You don't need much to actually do this.
And what about the composition of the films?
So the composition of films are, again, for the most part, approved products that are already on the market. So your sugars, your surfactants, and some polymer bases as well.
Mm-hmm. And have you found that the formulation of the film makes a significant difference to either the stability or the effectiveness of the delivery system?
It certainly does, and what we've really found too is there isn't one size fits all. I mean, we have to do a little bit of adapting and modifying based on the particular biologic that we're working on. But what is nice about that is we did a lot of the legwork initially. We screened so many formulations that we can pretty much predict what will and won't work within a reasonable amount of time, and then generate a film matrix that will work for a particular product.
And so you showed a couple of different examples of types of systems that could be loaded into these films effectively. Do you think that alternative delivery systems like the lipid nanoparticles or other delivery vehicles for genetic materials could be incorporated into this? And would the same type of stability profile be seen?
Yeah, and that's, like I said, I really do think we can adapt this to whatever type of biologic that we encounter. And so we've really started to do some work within LMPs to look at those things. And I think that, towards the end, I kind of wanted to highlight that is it definitely isn't a one matrix fits all, but we definitely are able to modify that to really accommodate it. And I think, what really accounts for the stability of this too is sometimes people look at it and they're like, you create the film and then you just spray the virus on it. It's really not that. It really has to be, the biologic has to be embedded and mixed within that film to really get it to work.
Yeah. And so the just a follow-up question, that the stability profile that you investigated that showed the excipients were binding with the hexon proteins, that's very specific to that formulation and that system. Are you seeing similar types of things, or have you studied similar types of things for other types of biologics, or how would you adapt your formulations to maintain that type of interaction and stability?
We are starting to branch out and really, because initially it was just like, let's see if we can stabilize this. And so we've successfully gone that part that we're really, now trying to do a more deeper down, dive into what is the mechanism. And so for the most part, that does seem to be with the virus capsids that, we do have materials that bind specific regions of the capsids that really seem to form, for lack of a better word, a protective shell or bubble around them.
Okay. So in one of your slides, you had talked about where the different formulations for the virus had different transport through the tissue in your in vitro tests. And can you speak a little bit to what aspects of the formulation or the system might be leading to those differences in transport?
Actually, the differences in transport that I showed you were with the same formulation, but we used different doses. So it was different, yeah. So we saw a dose effect, which was kind of nice, but we didn't really, at least in that particular study, I was just showing the impact of our optimized formulation that we had used in primates and everything else. And it was like, is it even going to get through the human buccal mucosa? And so it will, and we were kind of very excited about that, for sure.
Cool. Is there an equivalent immune tissue to the NALT in the sublingual buccal tissues that make the oral cavity capable of promoting the strong response that you saw?
So that's a very good question, and that's something we're actually investigating now with a few collaborators at the Tulane Primate Research Center to really understand, and that's something that's been largely unexplored. I've found as I've gone through, the literature, no one talks about the lymph nodes and what really populates around the mouth, and so we're looking at characterizing those and understanding the differences, if there are differences between rodents and primates, before we actually kind of move into humans for that particular route. So that's something that is under investigation.
So there was a question about some of the data you presented that was focused on the injectable or using these for injectable formulations. And so the question was around the viscosity, the 100 CP viscosity seems high for an injection using a syringe. Is there a strategy for managing that upon reconstitution and administration? I hinted to the fact that, especially with the very high viscosity formulation, we kind of did a 10x overfill. So you could dilute that out to something that was very workable. So, that's really our approach with the 100 centipoise as well, was to really, get it as low as we could get it and still form a film, but then we wouldn't have to address the 10x overfill. We could do something, much more reasonable. So. Given the composition of the films, is there any concern over like isotonicity in an injectable formulation?
We've basically just been, concerned about the viscosity of the full polymer solution as it is. What's nice is that we have done a lot of different rehydration studies with a variety of solutions and solvents. So there, is a variety of different things you can do to potentially address that. Because again, if you think about it, we've got a very concentrated, small surface area with a lot of things in it that you can address those osmolality, viscosity issues through rehydration techniques.
Okay. There's a question about controlling release rates of the viral systems from the films. Is that something that you've looked at, or have you identified whether or not there's a need to control the release rate for efficacy?
We haven't identified that there's necessarily a need, but we've also explored the fact that we can indeed, modify the matrix again without compromising stability to get different release patterns, something, rapid versus something. I think the data I showed you takes like 2 hours. So a lot of people are like, wow, 2 hours, somebody's going to have something in their mouth that long. And so we've modified it to be a much more rapid release as well.
Okay. Is the backing layer important for functional delivery, or is it just needed for the mechanical stability of the system in order to be able to peel it and apply it?
Well, they're really, at this point, we have no backing system when we do stability, so it's just simply naked film, I guess you could say, packaged and put in particular conditions to look at storage. But we do believe humans are humans, and people will want to take that film and swallow it, just because that's kind of what it looks like you want to do. And so the backing would be important for that, to prevent any kind of swallowing and create a unidirectional flow of the film. And I just kind of want to say too that when we did the mouse in vivo studies, those films dissolved quick. I mean, almost to the point that we thought we lost them because they're so small, but they really did dissolve quick, and there was really no, they woke up and didn't spit anything out. So I was excited about that.
So you talked a little bit about Gerada, the company that's being started to kind of advance this towards the clinic, but what are the technical next steps for you to move this towards a patient population?
So there are a few. I mean, one issue is scale up, and everybody's like, this is great that you can do this in your lab in Texas, but can you make 50,000 doses a day? And so we're actually talking with some partners now where, I hope by the end of the year, we'll have a good sense of, how scalable this actually is. And, with an oral dosage form, there's the whole issue of sterility too. So with an oral dosage form, you don't necessarily have as much stringent requirements, but we're also looking at interesting packaging options as well to maintain sterility and to do some neat things with regard to hydration of the films.
Mm-hmm. Do you think there's something specific about the film architecture itself that's leading to either performance or stability, or is it simply a matter of entrapping the materials into a matrix that's in the solid state?
I think it's important to trap traps the things within the matrix, but I also think, and this is a paper that's under review right now, actually in the journal control release, but we do have some data that really does show that some of these components, particularly a surfactant, can really bind these viruses in a matrix that really prevents them from being exposed to the environment.
Okay. Just making sure I got everyone's.
I'm trying to look too, so.
The questions keep coming. So have you considered spray drying for this technology?
We have not, only because at least with, early technologies, it just seemed like a harsh route to go. And so that's why we, to be perfectly honest, it was kind of luck at first. We're just like, let's try this and see what happens just at ambient temperature and go with it. And just to kind of refer back to the company, Dorada is a place, actually it's a city in Poland. It's a beachfront area that is the resource of most of the amber of the world. And so we think that this is our amber where we preserve live biologics within it. And so it's a little cheesy, but it works.
I like it, yeah. Do you have any thoughts around how you might be able to use these films for pediatric use, or how appropriate they would be for pediatrics?
I think they would be appropriate for pediatrics. And this is kind of, I guess, a side note. I actually have funding from the NIH, grants titled Cinnamon and Spices, Adjuvants Are Nice, that we're actually looking at the science of flavoring and how we can potentially use some of those flavorings at certain concentrations to be natural adjuvants. And we have some very nice cytokine responses in buccal explants and some things like that as well. So yes, these will taste good, but maybe not too good.
Yeah, and we only have about a minute left, but this kind of goes back to what we were just asking about with spray drying and sort of the film structure. But is the formulation still effective if you took the film and broke it up into particles?
So particles, I'm not necessarily sure about, but we have done, because we really toyed with the idea of, does it make more sense to make unit dose films, or does it make more sense to make sheets of these things and then cut them down into individual doses? And when we did do those large sheet experiments, we did see that we got even distribution of active virus throughout, from the first corner of the film to the last corner of the film. So that's possible, but we're still, toying with the idea, does it make economical sense? Because we may still have loss of vaccine with pieces that break off or something like that versus a single fill and just go that particular route.
Yeah, so one more question and then we'll be done. Is there any sort of mucoadhesive properties of the film, or is it just the rehydration process that keeps it in place when you administer it?
It definitely has some mucoadhesive properties for sure. I don't envision how it could work without that.
Okay. Great. Well, thank you so much for your time, Maria, and for the great presentation and answering all of our questions. We're going to pivot over now to the career panel. And so I just want to thank everyone who joined us this morning and also on Monday for participating in this event. And we're really excited for our in-person conference in 2023. So we hope to keep in touch with everyone and then see you in person at that point. So just remember to keep checking on the website and looking for communications as we get closer to those dates so that you can join us. And so I will hand it over to Leah and to Matt, who will introduce the panel participants and moderate that session. Thanks, everyone.
Hello, everyone.
Hi, everyone. Thank you so much, Erica, for the introduction. So we're moving on to the career panel portion of our meeting today. As Erica mentioned, my name is Leah Burson, and I'm here with Matt DeFries. We're the GRS co-chairs for 2023, and the GRS is part of the GRC that's targeted towards graduate students, early career scientists, postdocs, and we're excited for the career panel that we have planned for you today. We have 3 wonderful panelists that will be joining us, and they have a diverse set of knowledge and experience that they're going to share. I'll first briefly introduce each of the panelists, and then I'll ask them to give more of a detailed overview of their career path and what their current position is. During this time, feel free to ask questions in the Q&A function of the chat feature. And if you see a question that you particularly enjoy, make sure you give it an upvote so that it moves to the top and we make sure we get to it. So let's get started. Our first career panelist is Dr. Blair Bretman. That worked out perfectly with her appearing on screen. She is an assistant professor of chemical and biomolecular engineering and material science and engineering at Georgia Tech. And Blair's research focuses on linking molecular to micron scale phenomena to processing and multi-component complex mixtures to enable rapid and science-driven formulation and product development. Next, we have Dr. Jennifer McGuire, and she is the Deputy Director of the Office of Quality Surveillance, OPQ CDER at the FDA. And in her role in this office, she assesses the intelligence throughout the product lifecycle to inform stakeholders about the state of pharmaceutical quality, and she uses data analytics to drive surveillance decisions. Last but not least, we have Dr. Yuchuan Gong. He's the Vice President and Head of Small Molecule CMC at Beijing Limited, and his responsibilities include new chemical entity development and clinical manufacturing. And throughout his time in industry, he's gained experience in small molecule product development and commercialization. So just first, thank you all for joining us today for this career panel. We're excited to have you here. And now I'd just like to ask each of you to give a short 2 to 3 minute introduction about your career path and what you're doing in your current role so that we can get to know you a little bit better. So we'll start with Blair.
Hi. Yeah, thank you very much for the invitation to participate in this. I am currently a professor in chemical and biomolecular engineering and material science and engineering. And before that, I did a postdoc at the University of Chicago in molecular engineering, focusing on polymer theory. And then before my postdoc, though, I actually worked in industry. And so, I did my PhD and I had no interest in ever being a professor. So I went to industry, and I was actually in the materials industry, not the pharma industry. But that's because my PhD was in manufacturing-based approaches, which are very translational. And so I worked for a company called Saint-Gobain for 2 years after my PhD, and then went into a postdoc. And then I guess kind of working backwards, my PhD was in chemical engineering at MIT as part of the MIT Novartis Center for Continuous Manufacturing of Pharmaceuticals. So looking at transitioning from batch to continuous, and really my focus was on the downstream side, paying attention to how formulation impacts processing as well as the product. And so, I've had a little bit of a windy career path. I've also done internships in nano fabrication, internships in a sugar refinery in India, all sorts of highly varied experiences, which really has led me to this nice career in academia. We'll probably get to it a little more. I think there's some questions about forks in the road coming up. But just briefly, my kind of decisions of heading back into academia from industry. It was a great company that I worked for, but I didn't feel like I got to go into a lot of depth on things. And so I sat and spent a lot of time reflecting on what values I had and what, different aspects of the day-to-day job I appreciated, and decided I wanted to be somewhere where I had a little bit more freedom to dive into the science, and be a little bit less commercialization focused. And so, lots of reflection on all of that. And so that led me to Georgia Tech, where it's been a great experience working with students and getting to do science and mentoring significantly. So, yep.
Thanks, Blair. I've jumped in a little bit in my short career path too, so I'm looking forward to those questions later and hearing your perspective. Next, let's have Jennifer. Could you give a brief overview of your career path so far?
Sure. I've actually pivoted a couple times myself. So I started with a degree in chemical engineering, and at the time that I graduated, I was pretty burned out. So the thought of graduate school was not even in the realm of possibilities. So I went ahead and took a job in industry, initially doing API synthesis and scale up. And I really enjoyed the pilot plant aspect, not so much being in a lab, but ultimately got to the point where I wanted to do something a little bit closer to the patient. So I took a position with another pharma company focused on formulation and pharmaceutical finished dosage form manufacturing, and really enjoyed that. But after about 5 years or so in industry, I did realize that if you're in the research environment, there can be a bit of a glass ceiling if you don't have a doctorate. So I made the really hard decision of quitting my nice paying job and relocating my husband and me to the Midwest so I could go back to graduate school. And in graduate school, I took another slight pivot because I ended up studying under a professor that was doing vaccine formulation with aluminum containing adjuvants. So I had in my head that I would be focused on solid oral dosage forms because that's what I knew. But, another slight pivot. And then after graduate school, I graduated at a challenging time with the economy, and the job opportunity that presented itself was actually with the Food and Drug Administration. So I came into the FDA thinking that I was on a 3 to 5 year plan, get a little exposure to the regulatory environment, and then head back to industry. That didn't happen. So I've actually enjoyed my time at the agency. I just hit 10 years with FDA. So I started as a reviewer reviewing generic drug applications and then eventually new drug applications as well, reviewing the chemistry, manufacturing, and control information. I spent a number of years in a review office, but more recently have pivoted over to a leadership position in our Office of Quality Surveillance. And so it's a new office, and the way I would describe it is it's kind of like a mashup between the CIA and the NSA for drug quality. So we're surveilling the market and looking for early signals that indicate that a drug might have manufacturing problems or things of that nature, and really being able to understand the state of pharmaceutical quality. So that's, a brief overview of my career, and I'm looking forward to questions later.
Thank you so much, Jennifer. We also have a similar career path in that I went to industry and then ended up back at Purdue in IPPH, which is where I am now. So it's nice to see another Purdue person here on screen. And next, last but not least, we have Yu Chuan Gong. Could you give us a brief overview of your career so far?
Sure. Can you hear me all right?
Yeah.
Yeah, great. Thanks for inviting me. So it's a pleasure to be part of this event. So I did my PhD at the University of Puerto Rico in physical chemistry. Actually, I got into the field of pharmaceutical science by doing a postdoc with David Grant at the University of Minnesota. So over there, I actually started my academia career briefly. And then I realized that I'm still that type of person that I wanted to do develop some product. So just at least I want to give it a try. That's why I joined Vertex again for briefly a year. So developed a team to do the solid form screening. So over there, I realized that I knew science, but I did not know that much of like the entire industry, how people work together to develop a drug product. So I grabbed the opportunity to move to Abbott at the time, later on become AbbVie. So over there, I got a lot of support from my management, allowed me to go from beginning of development from this API crystallization, a solid form, then pre-formulation, formulation, then even give me opportunity to work on the commercialization of a drug product. So I spent 11 years over there before I moved to Celgene to head of the formulation group over there. So over there, I applied all the knowledge I learned over years and also applied some of my own thinking on it to try to make it more efficient. And I'm pretty satisfied with what I did at Celgene. Then later on, Celgene got merged with the BMS. I kind of wanted to find another opportunity to work in biotech. That's why 3 months ago, I left the BMS and joined the Beijing. So now I'm the head of CMC, a small molecule CMC, in charge of the analytical drug substance, drug product, and clinical manufacturing. So yeah, that's pretty much my career path. And then I'm looking forward to have more discussion with everyone here.
Great. It sounds like you've had a very exciting career thus far. I'm now going to hand control over to Matt, who's going to start off with some questions for you guys. And everyone in the audience, as a reminder, pop some questions into the Q&A and we'll get them answered for you.
Thank you, Leah, and thank you guys for those introductions on your background. That was great. I would just to quickly introduce myself and echo some of those comments. I likewise did chemical engineering, worked at industry, returned for my PhD, so it seems to be a common theme. So I think, you know, if you're new to this industry, you're a student and you find yourself taking a lot of twists and turns, it seems to be part of the course. So that's good to hear. And I just recently graduated my PhD and I joined Catalent as a development scientist for late stage process and product development. Okay, so the goal of this was to kind of cover some questions that the audience had, as well as maybe get an idea of your kind of past, present, and future, how you're structuring your career and your growth. I think we've covered the past section very well. From some of the questions that have come in so far, probably it looks like the one getting most attention is, so what's your guys' advice for building and maintaining a network? Let's start with you, Sean, if we can.
Well, thanks. So this is a good question. Network definitely is a very, very, very important element in our career development. I would say that we do need the mentors, and we do need peers who can help us out. I think that the most important thing is that we do need to know what we need, right? So what we want to, where we want to improve ourselves, what kind of extra knowledge we want to get, and so then you plan this out. I mean, you have a goal, then you have to reach out to people, then you have to keep connected. Basically, it means that, for example, like my own mentor, I still keep like talking to her like every month, even we are in 2 different companies. Right? So just stay connected, always give them an update, and then ask questions.
I would echo that. I would say I've also, like I send emails regularly to the advisors I've had in the past on an annual basis. I find that helps a lot as well to stay connected and engaged with people. So that's a good point.
I have a follow-up question on that. So maybe both of you, Matt, since you said you send these yearly emails, do you send them with pointed questions, or just it's really, hey, I just want to tell you about what's going on with me because I think that you're interested. I feel like maybe I need to have a reason to reach out to these people to maintain these connections, and sometimes find like, oh, I don't have anything to ask, or I don't need anything from them, so I'll just keep quiet.
I keep it covered. Oh, it's Christmas time, just seeing how you doing and giving up. That's what I do.
Yeah.
If anyone else is.
Yeah, I tend to do the same thing, that I want to do over time. I turn this relationship more of to the personal relationship, right? And I feel more comfortable to talk about anything, not necessarily a specific question. Right?
Okay, thanks.
Yeah, so I have something to add kind of to the first, I guess, the question about networking. So I had a kind of mentoring meeting with a bunch of graduate students recently, and we were talking about it. And I think people think of it as networking, but I think it's a parallel path of visibility in networking, because the more you can get yourself visibility in your professional spaces. So for academia, that's really a very broad space. It's, faculty throughout the country. For companies, it can be your management, different depending on your management structure, but most companies, you should be trying to get some visibility. And so the more you get visibility, the easier it is to build and maintain a network. To get more visibility, then you have to use your network. So there's these parallel paths where you kind of slowly make your way up, kind of trading off. And, I learned a lot of this actually from my boss in industry, who recognized the need for visibility for his employees and his group, and really helped us get these small opportunities that had upper management learn who we were. And then once we had that, the networking got easier and all that. And so I think that's really how it works in every field. The details of it varies a lot, depending on academia, industry, and I would guess also, national labs and other spaces. But I think if you think of it less about, okay, I need to find this network and remember this single person, and more about combining just being visible with some specific people, I think that's a really successful way to move forward there.
Yeah.
I think it's a good point.
Yeah, I think that's a good point. And I would say networking sometimes can feel uncomfortable if I'm actually, surprisingly shy, but going to those networking events that are associated with a lot of conferences end up being really beneficial. I would say that the pharma industry overall, believe it or not, is pretty small. And people move around between companies. So over time, you will start to recognize people, and they'll know people that you know because they worked together at a previous company. And it does become slightly more natural to kind of engage with people and talk about what's going on. And so really leveraging those networking opportunities associated with conferences that you may attend is a good way to kind of start building your network.
Yeah, and I would echo, we have a, open networking session after this. So, stick around at a table, I'll stick around, just introduce yourself, get some practice talking to people. It's, practice makes perfect, so for what it's worth.
Yeah.
So I think we have a lot of interest in this area, and we talked about building a network, but how do we find maybe a more personal relationship in terms of developing a mentorship relationship with someone? How do you go about finding a mentor and make the most of that relationship? Maybe Blair, do you have a comment?
Yeah, so we talk about this a lot with academia and faculty because it's very important to have sponsors and mentors to kind of work your way up. And unfortunately, we haven't found an easy solution. My personal preference has become get someone formal right away whenever you're somewhere new, whether you click with them well or not, just get something going for your first year somewhere so you can get all the stupid questions answered. And then start talking to more and more people and find the people who you naturally trust, right? Because it is really important in a mentoring relationship, not just to have someone who can help you, but it also has to be someone who you can develop trust with. And that can actually take quite a bit of time. So most of my mentors tend to be people who, fit well with my personality and that they're very strategic. They like to think things through a lot. They never tell me what to do. They're the type that like to ask questions, but that fits well with my personality. And if, I try to find mentors just based on their kind of career level or something like that, it may not work well because we don't develop that rapport. But I do think it's still important to have that initial person wherever you're somewhere new or in a new position, just to get you kicked off. And then really just working on talking to as many people as you can until you find a good natural fit.
Thank you. Yeah. Anyone else have comment on that?
Yeah, I would say another aspect of mentoring. So in addition to having more of those formal relationships that you establish with someone to kind of bounce ideas and get a little direction and questions answered, one of the things that I've also found helpful is as I've moved through my career and had aspirations of moving into leadership, looking at the different leaders in my organization and seeing what behaviors they exhibited that I wanted to, on some level, emulate. So to give you an example, at times at work, meetings can become very controversial and sometimes tempers escalate, but certain leaders are very, very good at maintaining their composure and just kind of keeping things on track. So that's something that I was interested in, like honing that skill and learning, how did that person master it? Because it's not easy and it doesn't always come natural. It's something that people work towards. So setting up a time to talk to that person and be like, hey, this is something that I'm working on. I noticed you did a really good job in that meeting of keeping your composure. What is it that kind of led you to hone that skill? Or what are you telling yourself to kind of keep calm, cool, and collected? So just sometimes looking for people that are doing something that you aspire to do and then having a conversation with them about how they got to that place is really helpful.
Yeah. So I would echo those sentiments. I think Blair kind of mentioned where she chased her values in terms of going back into academics, but I think that works with your mentors as well. Find someone who really exemplifies the values that you hold dear and reach out and connect with them. And if you do hold those values dear and they do as well, then they're going to recognize that in you, and they'll be probably very open to developing a relationship. So maybe it's a little awkward. You don't just ask, can I be, my mentor? But I remember a professor who's really rigorous, very fundamental mathematics and solving complex issues. And I just love that. That's one of the reasons I went back to grad school. And Dr. Bradley Anderson, and I still cherish the advice and time and conversations I've had with him. But you just got to develop the relationship, introduce yourself, and reach out. So that's my recommendation. But thank you very much.
I have a little practical advice actually to add from my time in industry. When someone joined after me, they received advice to take each person in the group out to lunch, or maybe not take them out to lunch, but ask them to go to one-on-one lunch, maybe either in the cafeteria or out to a restaurant. And he slowly went through everyone in the group. And I thought it was really nice personally that I got to have one-on-one time with him. But for him, he got to have this one-on-one time with everyone in the group. And through that could probably find these things that Blair and Jennifer were talking about, different personality traits or different skills that they have that they would want to work on. And then they could use those people as a mentor through this kind of scanning of the department that they joined. So I thought that was a great idea and hope to use that in the future.
I get the same advice for when I started my faculty job. As they said, take all your colleagues out to lunch at some point over the first few months. So it seems to be a very common, successful strategy.
Yeah,
It's a good idea.
Yeah, a comment I want to add on that is that for anyone who wants to find a mentor or mentors, so just don't be afraid. I found out that a lot of us just feel kind of nervous, right? So you want to have someone senior as your mentor, then you always find that that person has a high rank or the person is very busy. Just don't worry about it. The more successful a person is, the more willing they are sharing their experiences. Just reach out and ask them. So you'll be quite surprised.
Yeah, I agree. Those are very good points. Thank you.
Right.
So the next question that's getting attention is, it seems there's a lot of moving between companies in the pharma field. Do you think it's necessary to move up the ladder, or is it possible to stay at the same company for a long time? Does anyone like to jump on that? Maybe you, John, since you work in industry.
I knew that this is coming to me. So you can obviously see that I spent 11 years at AbbVie. Before I moved on to Celgene. I worked at Celgene for 3 years, and now I'm part of Beijing. So I would say that it has they're different. So it depends on what you need. At the beginning of my career, I wanted to learn how to develop a drug product from beginning to the end. Right, that's the reason why I wanted to join Abbott. Then when you work with a company like that at Abbott, well, I'm sure that at Merck or Pfizer or this kind of company, they have expertise at each of those functions. So they have people with years of experience. So that's where you can build up your technical skill, right? So I would say that in that case, it definitely worth to stay in a company like that for years and before you feel ready to move on. But then it's just me. I'm not saying that this is, should be the only way to build our career, because it depends on what exactly you want, right? A lot of people wants to be more like in the management side. It doesn't matter it's a technical management or it's like a supply chain management. In that case, you might want to find out that you might look for some opportunities to give you that opportunity to do things. I mean, in general, I would say that the title is not the most important thing. What you can gain, the knowledge and the experience you gain from that position is what we should look for, right?
Yeah, I agree entirely. And so maybe what taking from those comments is, when you know it's time to move on to your next step is maybe when, you're not growing in that role anymore. And that may or may not mean leaving the company, but it may mean identifying a new role for yourself to continue growing.
The question is still to me?
No, I was commenting and saying that I think I was trying to put it back in words in terms of, you don't have to leave, but, you have these opportunities to grow in a role and you stay there until you've reached your cap in terms of your personal growth as one way of making that decision. Yeah. Thank you. Yeah. Okay. So we also have questions coming in about the pandemic and what impact has that had on your field? What would your advice be for grad students, postdocs, early career scientists in this field? Maybe Jennifer, you might have a good comment since maybe at the FDA you kind of see a broad impact. I don't know. But.
Yeah, I can say so we've been 100% remote now since March of 2020. And so the commute's been great. One of the things that has happened though is like the work day has expanded. So, and it's meeting after meeting after meeting because everything is virtual. But I think that the agency has learned that we can be remote and still be very productive. So we're hopeful in the future that some of the flexibilities around teleworking will be expanded, although we already had a pretty liberal work at home policy. There are people though that don't have work that's portable, right? So the agency actually has a research and lab function, and so obviously those folks, had to get back on site. But yeah, I think it's definitely been a challenge. I was giggling before because I'm trying to be very focused and my 8-year-old just busts into the room and it's summer vacation. And so she's like, "What are you doing, mommy?" So there's those challenges that I think all of us have been managing. But by and large, I think, we've been pretty good as an industry overall at being productive virtually.
That's good. Anyone else have any other comments on that?
Yeah, I mean, I can talk to academia a little bit. I'm sure a lot of people know from the student perspective, but maybe less from the faculty perspective. The biggest change for us this year was teaching, having to do teaching hybrid or at our university or some virtual, which about doubles the amount of time we have to spend on it for most people. And hybrid teaching is just miserable. And so there's a lot of discussion about how we manage going forward. What elements of that stay? What is teaching going to look like in the future? I think from a research perspective, once people were back in the lab, a lot of stuff didn't change too much. There's more virtual meetings instead of in-person meetings. But I think my students spend a lot more time reading the literature, which they should have done before. So in general, there's maybe some positives there too about having more work from home time. But I think the biggest changes in academia are really going to be on the teaching and administrative side, and really how that, the potential for remote work, the dollar signs they're seeing in potential distance learning, and how all of that is going to affect it. So, as someone who likes to reevaluate my career all the time, I do think about, okay, if teaching is going to become double the amount of work it was before, am I going to stay in this field? And so I think it'll be interesting in the next few years to kind of see how things develop in that way. But I think on the academic side, a lot more has to do with teaching and administration.
Yeah, that makes sense. I would comment from my experience, at least wrapping up my PhD, the pandemic was strangely helpful because it forced me out of the lab and got some writing done. So, so that was actually helpful for what that's worth. Okay, so we have more questions about mentorship, and I think building a relationship with a mentor is reaching out to a mentor via LinkedIn. Is this a good idea or not? And what are your thoughts? Maybe you, Sean.
All right. Okay. Yeah. So thank you. I never did that, and so it's more because I feel less comfortable to ask for a mentorship behind the screen, because you are not really presenting yourself to the other party. Then in that case, you put yourself in the other person's shoes, right? How does that person know who you are? So I have never done that. No.
I would agree, and I've received, I don't know, once you have a LinkedIn account and tend to get a lot of messages from things, it's hard to screen the good from the bad. And I think LinkedIn's a tool to maintain your relationships. It's not one maybe to initiate. That's my
Yeah, I'll say there's maybe one exception where I found LinkedIn people have been successful with reaching out to me that I haven't met is, because I've done the industry back to academia and like industry post PhD, I've had a number of people who hear about that through someone I know, find me on LinkedIn and reach out to me that way. So when it's something very specific and you have a connection in common, I think it can be a nice tool to do that rather than like an email, because then I immediately see who the connection is and kind of what we have in common. But it only really works for me if it's these highly specific things in that case.
Yeah, that's a good point. I would agree with that. Make some connection if you are going to go that route so that they have to.
Yep.
So, all right. So do we have any advice for students trying to get started in their career in the industry? Maybe Jennifer or Blair? I feel like I've asked you, Sean, that question already.
I mean, as someone who left industry, I have some advice for how you can do it better. So maybe I'll start us off.
Yeah,
so I was desperate to leave my PhD and finish, as probably many people are. And I, kind of vaguely knew about industry from internships, but it's very different in kind of real industry once you're there full time. And so I was very ill prepared for what day-to-day industry would look like, for the fact that you no longer have any goals, right? So you're staring down the barrel of a 30 to 40 year career with no milestones anymore, compared to getting your undergrad degree, getting your master's, getting your PhD, where you always have clear milestones. And so I think the advice I would have if you want to go into industry is talk to people who have been there longer, not just fresh graduates, and really hear about different career paths and different opportunities, so that when you start, you can start defining kind of some moderate goals, right? Okay, within the first few years, I'd like to do this, even if it completely changes. But that huge jump from being in academia and getting your degrees to suddenly having nothing, going forward can be really hard. And I think a lot of students are ill prepared for that, and when they first start their careers. And so I think really spending a lot of time thinking through what you like, listening to more senior people in a variety of roles would be a very helpful thing.
Yeah.
I agree.
And I would say be open. You never know where the next opportunity is going to come from. So, it's one thing to have a career plan, but just be open to opportunities. So I certainly didn't think when I went to graduate school that I would do anything remotely related to vaccines. And then I was shocked when I went to the agency that I opted to go to an area that didn't really touch vaccines. So just being open to the opportunities that present themselves to you, and asking yourself, is it an opportunity that's going to stretch me? Is it going to challenge me? Am I going to learn something? Is it going to keep me interested? And if it does all of those things, then it might be okay, even if it's not what you thought or what you envisioned for yourself. So.
I have a follow-up question for you, Jennifer. Since you mentioned that you went into a position that was a totally different field than your PhD work, how did you leverage the skills that you had from your PhD to interview and to say, I can still do this job even though it's not what my research was based on during my PhD?
Right. So the biggest thing about kind of selling yourself after the PhD is it's not so much, it can be what you studied if you intend to stay in that area. But if you're open to change, it's not so much the exact subject that you studied, but it's all of the critical thinking skills and problem solving that you've learned. So I think, going through the process of getting my PhD very much impacted the way I attack problems and the way, a roadblock pops up and now I have to get around it. Like, what are the options? How do I think through it? How do I keep this on track? Those skills don't leave you, and they apply kind of globally to the work environment. So it's really selling those skills and showing how they relate that become very valuable.
Okay, another follow-up question. Since I'm late in my PhD, starting to look at jobs, and I think maybe other people in the audience are as well. When I look at job postings, there are all these details about the job that maybe are unrelated to what I know, but it doesn't, the job posting doesn't say we want a critical thinker who's good at roadblocks and those kind of skills that you just mentioned. So how do you know which job would be a good fit for you if you're using these maybe soft skills and not the technical knowledge to fit into a position?
Yeah, that's a good question. So I think it's hard to know until like you interview and you have the opportunity to talk to someone to figure out if it's a perfect match for what you're looking for. But I would say in terms of like marketing yourself, the power of the cover letter. So if you're applying to something that you on paper don't seem to be a fit for, but you think there's things in your experience that fit, making sure that you use the cover letter to kind of point out why it's potentially a good fit can be really helpful.
Okay, thank you. The dreaded cover letter.
Yeah.
I think I would, again, kind of echo too, or follow up on that, is to say that, like, you know, if you're not a good fit, maybe ask yourself, why do you want to apply for that job? Because I know when I was applying for a job, it's like, I want a job, just give me anything. What's happening, right? Okay, so you need to screen yourself a little bit. Be honest with yourself. Are you wasting your time even applying, not because you won't get an offer, but because it's not something you really want to do? So be a little discriminative. And I know that can be hard when you're like, I need something.
Mm-hmm.
But that's just my comment, because the situations I found myself when that was the case, it really wasn't a good fit. It was just like, oh, it's close and it's local and it's easy, but that's not why you should seek a position, in my opinion. Just my comment. Okay, so I think we got to that question. All right, so I think another important question, and it's probably been all up in the air recently with the pandemic, but is, what's the flexibility in terms of work-life balance that you've had in your career? Is your role, because I have a lot of questions around this too, because sometimes it's hard to balance work-life balances, especially with a high demanding job that can, I think, occur in this industry. So maybe you, John, if you'd like to comment on that, how you maintain work-life balance or what you can expect.
Yeah, this is a very, very, very good question. Hopefully my answer won't surprise a lot of people, right? So we've been hearing about this work-life balance all the time, right? I think it's your own choice. Actually, I was quite surprised that when I talked to one of my mentors, who is pretty high up in a company, she basically told me that there's no such thing as a work-life balance. So it really depends on what you want to do, right? So your life is always there, and you have to get job done, right? So I echo that in my career life. So I've been working very hard, and a lot of people call me like a workaholic, right? So I never complain about it. I feel I really enjoy that, right? So, but it doesn't necessarily mean that I don't have a life. So throughout my career, again, when I was in graduate school, I told my wife that I'll absolutely spend a day every week, right? One weekend day with her, as wherever she wants to go, I'll go with her, right? So I kept that promise. So, but a lot of times she doesn't need to go anywhere, so I just end up working as well. But that's to me, I would say that it's enough of work-life balance, right? So I wouldn't call it like I have to spend a lot of time not working, just sitting there, and then it means that I have a work-life balance. I think it's just absolutely depend on individual.
Yeah, I would agree with that. I would like to put to rest this myth that academics have a worse, higher workload and worse work-life balance, because any ambitious person anywhere ends up struggling with this. I think academics, it might be there is no break where you have less, whereas, first couple of years in industry, maybe it's you haven't hit management yet, it might be slightly less. But if you're ambitious, it ends up a struggle you have to work on, and it's very personal how you decide to balance your time. I think I like academia for my personality because it works better for me to have full control of my schedule. So I often essentially take Thursdays instead of a weekend day because I can run errands and do things that are where places are closed on the weekend. So as long as I'm not teaching, I can just do that and nobody stops me. Whereas the company I worked for in industry, at least, was much more rigid with that. And so for my personality style, that was a better fit. But I still, as with you, John, I have to be dedicated to, okay, I'm going to make sure that I take the time I need and do that. But it's absolutely manageable in academia as well. Maybe there's more peer pressure. I think academics complain a lot and like to kind of be showy about how hard things are. So there might be a little more peer pressure to stand up to, but I know many people who have very good balance and ability to do things as fits best for them in all fields.
Yeah.
Yeah. Oh, good.
I would agree with what everyone has said so far. Work-life balance is challenging, and especially if you're ambitious. But one of the nice things about the government is I'm definitely busy, and work-life balance generally is challenging. But if something happened or my kid got sick or there was an emergency, it's very, very flexible. So I can take my laptop with me wherever and tap into work. And if I have to shut down and check out because something has happened, it's a very, very supportive environment. And there are people that will step up to fill in while I'm dealing with an emergency. So it's nice that when I do need to step away, this environment makes it very possible to do that. I'd also say, though, it's I have very poor work-life balance, but my husband has great work-life balance. So together, we have family balance, right? So, he very much wants to be an individual contributor in his role. He also works for the government. He has a lot more flexibility. So he's able to kind of pick up the slack in ways that I can't. So between the two of us, in terms of being there for the kids and whatnot, it works out. But, those are decisions and conversations that sometimes need to happen with a significant other to make sure that overall, the family's getting what they need. Just another aspect of balancing it all when multiple people have careers.
Yeah, I think there's a lot of good support for families throughout, all of the types of career paths in STEM right now. There's more push. I do think it's harder if you don't have a traditional family and you want a life. It's not really still socially acceptable to take a life for things other than children. And so I think hopefully there's going to be movement and progress in that direction as well. But I do think there's been progress, at least on the traditional family style side of things.
And we're just about wrapping up. I think we're supposed to end probably now, but I'm going to add my comment to that just because I've recently started and my experiences in industry is just to say maybe two things. Is when you first start, work-life balance is probably not as good as it's going to be because there's usually a learning curve, right? And you got to get through that period of time before you're really comfortable and good at your job. Maybe 6 to 12 months, depends on, up to you and your progression. But that's my experience so far. So don't get too discouraged if it's really hard when you get started. But once you adapt and acclimate, probably the same as you did in graduate school, managing your course load, managing your lab time. But by year 3 or 4, you see you're just the guy's wasting time all the time, right? So you see that grad school. So my point being is give yourself some time to learn and adjust and to get that balance. But it is certainly possible. And then I probably echo Yuchan's statements where work is my life because I enjoy it, so if I go get lost reading papers for 6 hours, that's my balance. I'm okay, but then I have to dedicate time. Otherwise I get in trouble. So anyhow, I think that was I think we're about done, right, Leah?
Yeah,
Okay.
I think we should wrap up now. Thank you all for your insight. It was really valuable. I definitely gained a lot from it, and I think our audience members did too. So thank you for participating with us today. For our audience members, we're going to have There's 30 minutes left, about 30 minutes. The platform will be open until noon. So this is an open networking session in that table format that you saw at the beginning of the presentation. So feel free to stick around. Open network for our career panelists if you have time. I don't want to mess up your work-life balance, but if you have time, it would be great if you could stick around, pop yourself at a table, and if anyone has some personal questions, they can come say hi. That wraps up the scheduled portion of our meeting. As Erica mentioned, we are looking forward to seeing the GRC community in 2023 in person. And keep an eye out in your email or on the GRC website for more information about that as things start to get planned for the future. And thank you again for attending today. And if you attended on Monday for that too, we're so happy to have had you here.
Thank you, everyone.